Hello, this is Remedial Biology, the revised version. In this video, we will see about protein synthesis. Under this topic, we will see functions of proteins, types of RNA and their roles in protein synthesis, the genetic code, transcription, translation, and the sites of protein synthesis. This video is prepared for remedial and grade 12 students. I am Dr. Alamayo from Warabe University. Stay with me.
To protein. If you see this diagram, you know that the monomer of protein is amino acid. Different amino acid joined together or bonded together by the peptide bond forms polypeptide. Now, if you take this is a single amino acid, this single amino acid can attach with another amino acid and they can form a chain of amino acid. Now, this chain of amino acid can form a protein molecules. Now, how this can be formed? Let us see the process. Proteins. Proteins are biological molecules which is composed of carbon, hydrogen, oxygen, nitrogen. Sometimes contains phosphorus and sulfur. Here, proteins made up of amino acid monomers. Here, amino acids general formula can have the R group, the carbon, hydrogen and the amine group, as well the carboxylic acid group. We will see that diagrammatically. Amino acids. Amino acids have a central carbon atom which is bonded to amine group and the carboxylic acid group. This is the central carbon atom for amino acid. In this central carbon atom, the carboxylic group is attached to this central carbon atom. Again, the amino group or the amine group is attached to this central carbon. And the other one is the R group is attached to this central carbon. Here, this R group this group varies in different amino acids. Therefore, it is known as R group or side chains. As the result, one amino acid can be different or can be vary from another amino acid depending upon its R group. These are some examples of amino acids. This one is valine. This one is lysine, aspartate, and alanine. Here, the R group in valine contains 3 carbon atoms. The R group in lysine contains the 4 carbon atoms. And in aspartate, the R group contains 2 carbon atoms. But in alanine, it contains 1 carbon atom. Therefore, each amino acid varies depending upon their R group. The R group can differentiate one amino acid from another amino acid. Here, the peptide bond. Each amino acid is attached together by the covalent bond, which is so-called the peptide bond. Here, if you take this as a one amino acid, which has this R group, and again, this one is another amino acid, when they come together by the process of condensation, that means by removing one molecule of water, they can form a bond which is so-called the peptide bond. This peptide bond joins 2 amino acids together, but the process can be reversed by addition of water, which is called hydrolysis. Here, amino acid linked together by the loss of a molecule of water from and form a peptide bond. Here, what's the function of protein in our body? Important functions of protein in your body. The first one is growth and maintenance. Here, protein is required for growth and the maintenance of tissue. The other functions of protein is that causes biochemical reactions. For example, enzymes. Enzymes are proteins. We have seen in our previous lesson about the cellular respiration and the photosynthesis, but each of the serious reaction can be facilitated or catalyzed by different enzymes, but that enzymes are proteins. They are proteins. Therefore, proteins can cause biochemical reactions. For example, enzymes are proteins that allow key chemical reaction to take place within your body. Here, again, protein act as a messenger. Here, amino acid chain of virus linkages from protein and the peptide which make up several of your body hormones. These proteins can be hormones again, and they can transmit information between your cells, and they can transmit information between your tissue, and they can transmit information between your organs, because the hormones are again proteins. The fourth importance of protein is that it provides structure. Here, a class of protein known as the fibrous protein, which provide various parts of your body with the structure, strength, and elasticity. For example, if you take your skin, if you pinch your skin, it is somewhat elastic. This elasticity of your skin is from the protein which is so-called elastin, which is found in our skin. Therefore, protein can provide our body's structures to be elastic or to be get its strength. The other function of protein is that maintains proper pH. Here, protein acts as a buffer system. Buffer system means it can maintain the pH at the required pH range. Therefore, in our body, there is a fluid. The pH of our body fluid can be maintained by the proteins which are found in our body. Therefore, protein acts as a buffer system, helping your body maintain proper pH value of the blood and other body fluids. The other one is balances fluids. Our body is full of fluids. Now the balance in our body can be maintained by the protein again. Protein in your blood maintain the fluid balance between your blood and the surrounding tissue. This can be maintained again by the protein. The seventh one is boost immune health. Protein form the antibodies. Antibodies are proteins, where our body is invaded by disease-causing organisms, for example, microorganisms like bacteria. Our body starts to produce antibodies which attack these foreign materials. But this antibody is a protein. Therefore, a protein helps us to boost our immune health. Again, protein provides energy. Here, protein can serve as a valuable energy source. This is only in the situation of fasting. During fasting, if there is no intake of food for about 18 to 48 hours, and at the time of exhaustive exercise, and at the time of inadequate calorie intake, the protein which is found in our body starts to give us energy because it can be catabolized and enter into the process of cellular respiration and provides us ATP. The other important functions of protein is that transports and stores nutrients in our body. Some protein transport nutrients throughout your entire body. For example, if you take hemoglobin, which is found on our red blood cell, it carries oxygen and distributes throughout our body. Here, hemoglobin is a protein. And again, the substance transported by protein includes nutrients like vitamins, minerals, blood sugars, cholesterol, and oxygen can be transported by protein. For example, oxygen is transported by hemoglobin. And again, protein also store nutrients. One best example, the protein that store nutrients is that ferritin. Ferritin is a protein which is a storage protein which can store the mineral which is so-called iron. Now, let us look how protein is synthesized in our cell. Protein synthesis is the stepwise process of the production of different type of proteins from amino acids, because amino acid is a monomer. In the protein synthesis, it involves DNA, RNA. Here, RNA is of three types. They are the messengeral RNA, transferal RNA, and the ribosomal RNA. They are important in the protein synthesis. And again, the monomer of protein, which is amino acids. Again, different or various enzymes are important for the process of protein synthesis. The other one is one organelle or the factory of protein, which is so-called ribosome. Ribosome is the organelle which is found inside the cytoplasm of eukaryotic cell, and it is the center of protein factory, or it is the place where the protein is synthesized in our cell. Now let us see, starting from DNA, the importance of DNA in protein synthesis. Here, DNA stores genetic information used to produce different protein. One of the main function of DNA in protein synthesis is that the storage of genetic information, the information that is important for the protein synthesis. Here, the other one is the messengeral RNA or messenger RNA. This messenger RNA, this is somewhat the structure of messenger RNA. In this RNA, there is a nitrogen basis, which is nominated by the green one is adenine. And again, they have a nitrogen basis, guanine. Also, they contain uracil and the cytosine. And the other portion is made up of the ribose sugar and the phosphate group. Then, the messengeral RNA is a single strand, or it has a single strand structure. This single strand is uncoiled. It is a straight strand of nucleic acid. It is found in the nucleus, and also you can find this in the cytoplasm of the cell. It is made in the nucleus, contains the nitrogen bases adenine, guanine, cytosine, and the uracil. It has no thymine. This thymine is in DNA, not in RNA. Therefore, its main function, or the main function of messengeral RNA during the protein synthesis, is that copies DNA's instruction, the genetic information, or the instruction for the protein synthesis is given by DNA. But the information which is given by DNA can be copied by messengeral RNA and carries the copies of this information to ribosome. Ribosome is found in the cytoplasm, and the protein is synthesized in the ribosome. Therefore, the main function of messenger RNAs carries the information from the DNA, and it can pass this information to the site of protein synthesis, which is so-called ribosome. Messenger RNA's base sequence is translated into amino acid as acid sequences of the protein. 3 consecutive bases on messenger RNA is so-called codon. This is so-called codon. For example, this is one base, which is uracil. This is another base, which is adenine. This is again another base, which is adenine. Now, these are 3 consecutive bases. These 3 consecutive bases is so-called codon on messenger RNA. And there are 4 nitrogen bases on messenger RNA strands derived from the DNA, which encodes a specific amino acid. Here, the 3 consecutive nitrogen bases so-called the codon, but each codon encodes a specific amino acid for the protein synthesis. And this messenger RNA, if it completes its mission for the first turn, it can be reusable. You can use one messenger RNA again and again. There are only 20 naturally existing amino acids, but the number of possible amino acid combination is 64 triplets, because there is 4 nitrogen bases, but one codon contains 3 sequences of nitrogen bases. As the result, 4 the power of 3 will give us 64. Therefore, there are 64 triplets. Out of 64 codons, 3 codons are stop codons. That means, in the process of protein synthesis, if the process reaches on this stop codon, the synthesis of stop will stop or it cease. And again, out of the 64 codons, 1 codon is the initiator codon. The initiator codon. That means it starts the process of protein synthesis. Therefore, it is so-called start codon. AUG is the start codon. Whereas the codon which stops the process of protein synthesis are 3 in number. They are UAG, UAA, and the third one is UGA. During the process of protein synthesis, if they reach one of these 3 codons, the process of protein synthesis will stop. But to begin the process of protein synthesis, the initiator codon or the start codon is very important, which is represented by AUG. AUG. The genetic code. Here, if you consider this, in this column, it is the first letters U, C, A, G, but this side it is the second letter U, C, A, G, and this one is the third letter U, C, A, G. Now, the 3 codon encodes for the specific amino acids I've said. The 3 sequences of nitrogen bases forms a codon. 1 codon specifies or encodes for a specific amino acid. For example, let me start from the start or the initiator codon. The initiator codon, it encodes for the amino acid which is so-called methionine. Methionine is the amino acid. The formation of this amino acid is encoded by the initiator codon or start codon. The base sequence of this initiator codon is, the first letter is, look, A, the second letter is U, and the third letter is here G. Therefore, AUG forms the codon for methionine. From this table, what we can understand is that the one amino acid can be encoded by more than one codons. The formation of one amino acid can be encoded by more than one codon. For example, take phenylalanine. Here you can find phenylalanine in two places. One of the codon which encodes for phenylalanine is that uracil, and the third letter is uracil. This encodes phenylalanine. The second one, the first letter uracil, the second letter uracil, and the third one is C, which is cytosine. Now, uracil, and the cytosine again encodes for phenylalanine. Again, if you see another amino acid, in this case, threonine. Threonine, you can find from this table at 4 places. That means 4 different codon can encode for threonine. The first one is adenine. Here is the second letter, cytosine, and uracil encode for threonine. And again, adenine, uracil, and the cytosine again encodes for threonine. In this way, we can conclude that from this one amino acid can be encoded by one or more than one codon. Here, if you see the stop codons, we have 3 stop codons. The codons of stop codon is that uracil, adenine, and adenine. And for the second one, uracil, guanine, and adenine. For the third stop codon is that uracil, adenine, and guanine. This is how the codons can encode for different amino acids. Each codon specifies particular amino acids. For example, the amino acid tryptophan is encoded by codon TAG. Alanine encoded by the codon guanine, cytosine, and adenine, guanine, cytosine, and cytosine. Glycine is encoded by guanine, adenine, guanine, etc. for each 20 amino acids. Now, let me come to the second important type of RNA, which is transferal RNA or transfer RNA. The transfer RNA has a clover leaf shape. Here, look, this one is the shape of transferal RNA. This transfer RNA has the shape of clover leaf. At one end, it has the site at which amino acid is attached. Here, this is the 3' side of transferal RNA. At the 3' side of transferal RNA, the amino acid can be attached on this side. But at the opposite side, it has the anticodon. In messengeral RNA, there is a codon, but in transferal RNA, there is anticodon. This anticodon is complementary with that of the codon on messengeral RNA. Here, the transfer RNA is a single-strand molecules with attachment site of one end for the amino acid. That is, at the 3' end. This amino acid attachment site is the 3' end. Opposite end has 3 nucleotide base, which is so-called anticodon, which is complementary with that of the codon on messengeral RNA. Here, the main function of transferal RNA is that carries amino acid in the cytoplasm to the ribosome, the site of protein synthesis, which is ribosome. It can carry different amino acid for the synthesis of protein to the ribosome for the protein assembly. 3 bases on transfer RNA that are complementary to the codon on messenger RNA, which is so-called anticodon. For example, if the codon on messenger RNA is UUA, uracil adenine, the anticodon is AAU, that means adenine uracil. Therefore, it's complementary with that of the codon on messenger RNA. Once the transferal RNA starts to carry amino acid from the cytoplasm, it enters the ribosome and reads messenger RNA codon and links together correct sequences of amino acid to make the protein. Here, this is the messenger RNA. Now, in this messenger RNA, there is a base sequence which you call that the codon. But again, this is the transferal RNA. At one side, or the 3' side of messenger RNA, there is the site at which amino acid is attached. Then the transferal RNA brings the amino acid from the cytoplasm to the ribosome and reads the codon on messenger RNA. But the complementary sequence which is found on transferal RNA is that the anticodon. So you see, the 3 bases on anticodons are complementary to that of the 3 bases of the codon on messenger RNA. Example, codon ACU, anticodon UGA. Now, like that of messenger RNA, transferal RNA is also reusable. The third type of RNA is that ribosomal RNA. Ribosomal RNA it is globular shape, helps make up the structure of the ribosome. One of its function is that to make the structure of ribosome. Together with the protein, ribosomal RNA can make the large units and the small units of ribosome. Important organelle for the protein synthesis, ribosome. Ribosome are the site of translation. That means making polypeptide. That is the protein factor in our cell. Ribosomes are cytoplasmic organelles that translate the messenger RNA template into the polypeptide chain. The process of protein synthesis involves conversion of instructions and DNA into functional products, which is protein, through transcription and the translation. Here, in the process of protein synthesis, there is the process which takes place, which is transcription, and again, translation. Here, transcription means it is the conversion of DNA toward this RNA, whereas translation is that is from RNA to formation, from RNA to protein synthesis. Here, a reverse transcription is a process in which DNA molecule can be synthesized from RNA templates. If this one is the RNA, by the process of reverse transcription from the template RNA, the DNA can be again produced. Here, this whole process of biological information flow is so-called as the central dogma of life. Process one by one. The first one is transcription. Transcription is the process by which DNA is copied, or copying of DNA means transcription or transcribed to messenger RNA, which carries information needed for the protein synthesis. Here, transcription takes place in 2 broad steps. The first step of transcription is that formation of pre-messenger RNA or pre-messenger RNA. This can be takes place with the involvement of the enzyme which we call that RNA polymerase enzyme. The second step of the transcription is that RNA splicing. This means it is addition. RNA splicing means addition. Addition of pre-messenger RNA to produce the desired messenger RNA. In the first step, the pre-messengeral RNA formed, but the pre-messengeral RNA which is formed at the first step contains the parts which is not important for the protein synthesis. Therefore, in the second step, which is RNA splicing, it must be edited. The process is so-called RNA splicing. That is to get the desired messengeral RNA. The formation of pre-messenger RNA. The mechanism of transcription has parallel with that of DNA replication. As with DNA replication, the partial unwinding of double helix must occur before transcription can take place. And it is the RNA polymerase enzyme that can catalyze this process. At the beginning of transcription, like that of replication, the double helix DNA should be unwinded. This can be facilitated by the enzyme RNA polymerase. But unlike DNA replication, in which both strands is copied, in DNA replication, both strands is copied, unlike this, in transcription, only one strand is transcribed. Now, let us see that process. The strand that contains the gene is so-called the sense strand, while the complementary strand is the antisense strand. The messengeral RNA produced in transcription is a copy of the sense strand. Here, our objective is to get messengeral RNA, but the messengeral RNA which we are going to produce is that it is the exact copy of that of the sense strand. But it is the antisense strand that is transcribed. Even if the messengeral RNA produced is a copy of sense strand, it is transcribed from that of the antisense strand. Antisense strand is the copied one. In this case, the ribonucleoside triphosphates align along the antisense DNA strand during this process. The RNA polymerase enzyme joins the ribonucleotides together to form the pre-messengeral RNA molecule that is complementary to the region of the antisense DNA strand. Transcription ends when RNA polymerase enzyme reaches the triplet basis of that is read as stop codon or stop signal. Until they reach on the codon or stop codon, the transcription proceeds. But when they reach on stop codon, the process will stop. Here, the DNA molecules rewind to reform the double helix, and finally, the pre-messengeral RNA we can form it. The formation of pre-messengeral RNA. In this case, if you take this one is a DNA, as you know, the DNA is a double strand nucleic acid. This double strand nucleic acid has the sense strand, this one, the sense strand, and again it has antisense strand. Like that of DNA replication, the first stage of DNA transcription is unwinding. That means it starts to open up here, unwinding, and RNA synthesis starts. Then when they start to unwind, here the pre-messengeral RNA, which is yellow in color, starts to be formed, which is the exact copy of that the sense strand, but it is transcribed from this strand, which is from the antisense strand. This pre-messengeral RNA is transcribed from the antisense strand, but it is the copy of that of the sense strand. Finally, the pre-messengeral RNA is formed. Then after that, the DNA become rewound. Then it releases of pre-messengeral RNA. That means it returns to its original position. Then after, the second step will proceed. The second step is that RNA splicing. The pre-messengeral RNA formed contains interons and again exons. But interons are not required for protein synthesis. They are not required for protein synthesis. Here, this one is the pre-messengeral RNA. This black portion represents the exon, which is important for protein synthesis, and somewhat the one which looks like white is that interons. Interon is not required for the protein synthesis. Therefore, it has to be removed from this pre-messengeral RNA to get the desired messengeral RNA. The process of removing these interons from pre-messengeral RNA takes place by RNA splicing. Then the pre-messengeral RNA is chopped into up to remove the interons and creates messengeral RNA. The process is which is so-called RNA splicing. By RNA splicing, the portion of pre-messengeral RNA which is not important in the process of protein synthesis, which is interon, is removed or chopped out. The other process or steps in the protein synthesis is that translation. Translation is the synthesis of protein from building blocks of protein, which is amino acid, based on the genetic information instructed on messengeral RNA with the help of ribosomal RNA, transferal RNA, and enzyme. Here, the transfer RNA carries a specific amino acid from the cytoplasm. One of the functions of transferal RNA is to carry and transport the amino acid which is required for the process of protein synthesis to ribosome. Then each amino acid has its own special transferal RNA. For example, the transferal RNA for the amino acid phenylalanine is this transferal RNA. Therefore, each amino acid has its own special transferal RNA. Here, each amino acid is attached to its transferal RNA through the 3' of transferal RNA. During translation, the first step is that messengeral RNA carries information from DNA aligned on the ribosome in the cytoplasm, because ribosome is found in the cytoplasm. The second step, the ribosome attach onto messengeral RNA and let the transferal RNA loaded with a specific amino acid to enter it. The third one, transferal RNA with anticodon bring this amino acid from the cytoplasm to the ribosome. The fourth step, the anticodon on transferal RNA pairs with the codon of messengeral RNA on the ribosome. At the fifth stage, the information in messengeral RNA translated into protein with the help of ribosomal RNA. Then the polypeptide chain of amino acid will then form a protein. The steps of translation. The red ones to represent the ribosome, which is found inside the cytoplasm. It has the large subunits and the small subunits. The messengeral RNA, which carries information from the DNA, become enter into these ribosomes. But the transferal RNA carries amino acid from the cytoplasm and transport into this ribosome and puts these amino acids at different positions on messenger RNA by reading its complementary codon through its anticodon. Because here ribosome has two sites, which is the P site and the A sites, at which the transferal RNA can be attached for some time or for temporarily. The ribosome made up of a large and a small subunit, which is composed of 40% of RNA and 60% of protein. Here it has the attachment sites for transferal RNA, which is the P site and the A site. That is the P site and A site. A means amino acyl sites, which accepts the incoming aminoacylated transferal RNA. It accepts the incoming transferal RNA. The P site, which holds the transferal RNA with the nascent peptide chain. Here, this is the large subunit of the ribosome, and this one is the small subunit, and these are the P sites and A sites on the ribosome. Here, the messenger RNA inserted into the ribosome. Now, it is the turn of transferal RNA to bring the amino acid from the cytoplasm to attach on its complementary codon on messenger RNA. Here, the first step of translation is that initiation. That is, here, these are the codons on messenger RNA, AUG. This AUG is encodes for methionine, which is initiator codon. Then, here, the amino acid, the transferal RNA, brings the first amino acid and through its anticodon become attached with the codon on messenger RNA by the hydrogen bond, and this way the first amino acids become enter into the ribosome. The second transferal RNA again will come. Here, the second transferal RNA, it brings the second amino acid. Here, the second transferal RNA has the anticodon. Now, this anticodon become attached to the codon on messenger RNA. Then, here, the second transferal RNA become attached here. Then after, the second amino acid become enter into the ribosome. Now, these two amino acids has to be bonded together. Look, the first amino acid then bonded with that of the second amino acid by the help of peptide bond. Then the third transferal RNA will come. This is the third transferal RNA. It brings the third amino acid, and again to become, when the third transferal RNA enter into the ribosome, the first transferal RNA leave the ribosome, and that it becomes ready for another turn to bring another amino acid. Look, this one is the first transferal RNA leave the ribosome, then it is amino acid is attached on the second amino acid, then the third transferal RNA will bring that of the third amino acid, then it occupies its sites. Here is the third amino acid become attached, and it proceeds in this ways and bringing different amino acids together, and these amino acids attached together by the peptide bond. Here is the fourth one. When the fourth one is coming in, the second transferal RNA leaves the ribosome, then in this way the first amino acid, which is brought by the first transferal RNA, bonded with the second amino acid, and the second one is with the third amino acid, totally around 200 amino acids bonded together in this process, and they form the primary structure of the protein, which is the peptide structures. Then finally it reaches on the step of termination. Termination means when they reach on terminator or stop codon. Stop codon here they reach on UAG, which is one of the stop codon. After this there is no more addition of amino acid. The process of translation becomes stop. Then this is the primary structure of protein is formed in this way. The end product of the protein synthesis, here the end product of protein synthesis is the primary structure of protein. This primary structure of protein is the sequence of amino acid bonded together by the peptide bond. The shape of protein is determined by these primary structures or sequence of amino acid, but the sequence of amino acid in the protein is determined by the sequence of nucleotide in the gene or DNA. Here these are the sequences of amino acids. These sequences of amino acids are determined by sequences of nucleotides in the gene, and they form the primary structures of protein. From this what we can conclude is that the primary structures of protein can be determined by the gene. Then the peptides, this type of polypeptides can be many type polypeptides can be formed, and they become polypeptides. The polypeptides come together and the form is the protein molecules. Here if you take this messenger RNA, the messenger RNA, the first codon is the start codon, which codes for methionine. The second codon, third, fourth, fifth, sixth, and the seventh. Then the first codon can bring this methionine, which is initiator, and the glycine, serine, isoleucine, glycine, and alanine, which are different type of amino acid. Now these sequences of amino acids are the primary structures of protein, which is bonded together by the peptide bond, and this structure is determined by the G. This is the end of this lesson on protein synthesis. Thank you very much.
Thank you for joining me today. If you enjoyed this lesson, hit the like button, subscribe, and share with your friends. Thank you. See you in the next video.